Plant pathogens are microbes that make contact and enter into a plant and cause disease. Pathogens infect plants because during their evolutionary development over millions of years, they have become able to subsist on those substances produced by their hosts, and some pathogens depend on these substances for survival in much the same way some mycorrhizae and rhizobia do. Of course, the main aspect that differentiates between mycorrhizal or rhizobial relationships from pathogenic ones is that the latter causes disease, physiological disorder. A microbe that exchanged, for example, nutritional substances with its host while simultaneously affecting some other aspect of its physiology enough to cause disease would still have some level of pathogenicity. Like, for example, if that pathogen maybe cut the ability for the plant to photosynthesize, or infected or necrotized certain parts of the cell, it would still cause disease and still be a disease pathogen, even if it had other beneficial effects. I think it is helpful to remember that these relationships between organisms are nuanced, and especially in the realm of phytopathology, an organism can be innocuous in one context and extremely pathogenic in another. Host specificity is a good example, as some pathogens have a broad range of hosts owed to a robust physiology that enables successful infection against different plant immune responses and ingress through different surfaces. There are two main areas where infection takes place: the phyllosphere and the rhizosphere. Pathogens that infect the foliage of plants first have to secure themselves to the plant in some way, and then enter into the plant. Before entrance, there are several barriers they must pass through, and there are a lot of novel adaptations pathogens have developed to neutralize both the physical and immunological defenses. For example, some fungal pathogens develop an aprosorium when their spores make contact with the host. That tendril-like appendage sprouts what's called a penetration peg, which must penetrate the waxy cuticle and other layers such as the pectin and cellulose lamellae. Cutinases, pectinases, and cellulases are produced that dissolve cutin, pectin, and cellulose, respectively. This is a popularly depicted method for pathogen entrance through foliage, but there are other routes. For example, Pseudomonas syringae, tomato DC3000, is a species of bacteria, or a pathovar, rather, that uses the virulence factor chronotine to actively open and then pass through the stomata, much to the chagrin and apoplectic dismay of cultivators around the world. Bypassing the waxy cuticle altogether, Clavibacter michiganensis, subspecies michiganensis, causes bacterial canker in tomatoes and can populate the surface of a leaf as an epiphyte before infection. Populations can be transferred through what's called guttation fluid, which originate from the xylem channels of plants and find themselves inside of a host in short order. Agrobacterium tumefaciens is related to other rhizobia, but instead of using its ability to interact with plants for their benefit, this species creates galls in them by inserting tumor-inducing plasmids. In the rhizosphere, the waxy cuticle is not present. In addition, roots produce barrier cells and root exudate containing metabolites and other byproducts, which have a range of effects on the rhizobiome, wherein microbes, both pathogenic and benign, exist. These effects can be toxic to some microbes, or they can be a source of sustenance. Some are chemical cues that stimulate microbes to make contact with the plant. In the case of arbuscular mycorrhizal fungi, these cues are important not only for initiating colonization, but genetic changes as well that occur in the fungus. These changes influence the plant host accommodating it at a genetic level as well. The cues are a lot like the Identify Friend or Foe IFF markers on some military hardware and vehicles. The use of these markers by microbes to sense and colonize the rhizobiome of a root system is also another way that pathogens can infect a host. The impetus for this video lies in the myriad questions that I receive, both in a personal and professional station, regarding plant physiology and plant defenses, namely, how to make plants even more defended against pathogens. And I think that I usually get questions that center around one or two different topics regarding pathogen defense, primarily whether or not a product, be it a biopesticide, a silicon amendment, or some other application that is meant to stimulate the immune response or the physical barriers that plants put up against pathogens, like the waxy cuticle on leaves, for example, or the turgidity and rigidity of plant cell walls, and things like this. And I think people sometimes misunderstand that even with these defenses, plants are still able to be infected in a variety of ways that will overmatch those defenses. And I give a bunch of evidence and examples in this video, and that's the reason why it is sort of short, because I want to articulate clearly that my understanding is that certain defenses are irrelevant regarding certain pathogen ingress, and it's not necessarily the case that a product that somebody applies will have any effect at all, especially in the case where a pest might vector a pathogen through, like, an aphid, for example. In any event, that's where I'm going to wrap. If you have any questions or comments about anything that I've said, please ask them and make them. I will include evidence for my claims in the description with some papers so you can do your own research and see where I'm pulling my information from. If in the future new information comes out and you would like to share it, please do. I am always interested in learning new information in the world of phytopathology. I certainly don't know everything there is to know. I don't think anyone really does. And I hope that this video has been helpful for you in your understanding of plant physiology. I will link videos at the end. Please check them out if you think that they will be helpful for you. Thank you for watching.